My name is Ruebel. I'm part of the computational bioscience group at Lawrence Berkeley National Lab. I will start off with, motivation and just a high-level introduction into what NWB is. I will then talk with you about different tools for converting your data to NWB, as well as tools for analyzing, visualizing, managing NWB data. And then towards the end, I will talk a little bit about extending the NWB data standard. As Steph already mentioned, this is really a multidisciplinary team effort, and I really encourage you to, as part of this event, to use it also to network with all the people that are attending the event and to get to know the NWB team. So what motivated the creation for NWB is, first of all, neurophysiology data are really expensive to collect, but at the same time, neuroscience data sets are very rich and can be used often to answer many different questions that are beyond what the initial intent of the experiment was. And so reusing data saves money and time and reduces also animal use. And so sharing neurophysiology data within a lab and with collaborators at the same time is hard. And so everybody has different ways of storing the data. Metadata is described in different ways. And so with NWB, we're seeking to address this challenge by harmonizing how we share data. And then finally, scientific results and analysis are often hard to reproduce and compare, right? Different processing and analysis visualization tools require different data formats, and so converting your data back and forth between different esoteric formats is costly. And by providing a common data standard, we can create a harmonized ecosystem of tools that can interact with each other and make our lives much easier. And by providing consistent metadata, it also enhances the interpretation of the data. But standardized neurophysiology data is hard. We're dealing with a large diversity of experiments and a high rate of innovation. We have a high diversity in both species and tasks, ranging from experiments in mice and rats to humans to worms, all across the spectrum. There's a broad range of data modalities, ranging from optical physiology to electrical probes to ECOG devices, simulation, or intracellular electrophysiology. And then each of those different modalities has their own ways of processing signals to, for example, detect spikes or detect neurons in imaging data, and then do subsequent data processing and extraction. So there's a very diverse range of different data modalities and types of experiments that we need to deal with. So what then is NWB? At its core, NWB is an ecosystem for neuroscience data standardization. And so at its most fundamental core, NWB defines a unified data standard for neurophysiology data that focuses on the dynamics of groups of neurons measured on a large range of experimental conditions. And so NWB supports storage of a broad range of measurements for neural activity, including extracellular, intracellular, and optical physiology, metadata about the data acquisition itself, such as the devices, information about the experimental design, such as the stimuli that were applied, documentation of the trial structure of experiments, and so on, as well as metadata about the subject, and then finally also information about the actual behavior of the animal through, for example, motion tracking and other forms of behavioral recordings. Importantly, NWB is an extensible data standard. So new data types and modalities can be integrated with NWB through formal extensions, so that if NWB does not cover, let's say, a particular portion of the experiment, you're able to create your own extensions to integrate that data then with NWB as well, so that you can share all the standardized portions of your experiments with the community very easily, while any custom portions of your experiments you can still share through the same data standard by creating your own extensions. And so at its core, when you look at NWB as a data standard, it is defined through a formal data schema that organizes the data in an NWB file. At a high level, NWB has a set of different groups or folders. There's the general folder, which contains all the high-level metadata, essentially all information that you can get before you actually run the experiment, goes into general. That's information about your subject, information about the experimenters that ran the experiment, or let's say in the case of extracellular electrophysiology, there is information about the electrodes that you're using for recording and information about the devices. The actual acquisition of, let's say in this case, electrical recordings, then is stored in the acquisition folder. For each modality and different type of data, we have what we call neurodata types. Each neurodata type identifies a particular kind of data, in this particular case, an electrical series. So a core concept in NWB is this notion of a time series. So these are recordings of signals over time. The first dimension in the time series is always time. And then you have additional dimensions for other data dimensions. So in the case of electrical recording, you have an electrical series that is time by electrodes. And then importantly, we always link back to the metadata that is relevant. So in this particular case, the electrodes will point back to our electrodes table that is in general to uniquely identify which electrodes you're actually referring to in this particular recording. For further processing of the data, for example, to extract spikes, that information then is stored in processing. And then for extracellular electrophysiology, then there is the units table, which is used for storing spike sorting results, such as then for each unit, you have information about the different times when a spike occurred, what the mean waveforms for those spikes are, which electrodes the spikes were observed on, and so on. And then there's also, for example, the intervals folder, which is what you're going to use to annotate intervals in time. And so NWB then provides a large set of these neurodata types for organizing different types of data. That includes neurodata types for extracellular electrophysiology, intercellular electrophysiology, optical physiology, behavior, stimuli, and then also experimental metadata. And so at its core, this is what you're going to learn this week are the different types that you need to use to store your data. And so through this, NWB enables unified storage of multimodal raw and processed data. So in this particular example, we have an experiment where the scientist acquires both optical physiology and electrophysiology data simultaneously, and both those recordings can go into the same NWB file. The data is then further processed. So the electrophysiology is processed through spike sorting to extract the units and their spikes, and then the optical physiology is processed to detect the individual neurons and extract the fluorescence traces. And again, that same information can go into the same NWB file. Importantly, in an NWB file, all signals are aligned to a common central reference time. And so with that, we can then visualize all these signals as a time synchronized trace next to each other very easily. So in this case, we have both the ePhys data and the DF over F from the optical recording, as well as an image of the actual neuron that the optical recording is showing. And so by synchronizing all these different signals in a single file, it makes analysis significantly easier. Beyond just defining a data standard, NWB provides a broad ecosystem of tools for neuroscience data standardization. That includes tools for data conversion and inspection that you will learn about during this workshop, such as Guide, NeuroConf, as well as the NWB Inspector. It includes our core data APIs in Python and MATLAB that allow you to flexibly interact with the data standard. It includes tools to define extensions and share extensions to the NWB data standard with the community, as well as then the actual core data schema, which is what you may think of as the reference for the data standard. And then finally, a set of core data modeling tools that a lot of our software built on, such as the hierarchical data modeling framework that defines our schema language and other tools that we build our Python APIs on. And so the first thing I want to introduce you to now are a range of different tools for conversion. So our goal is to enable researchers like you to effectively integrate your data with the NWB data standard. And so NWB by now provides a range of tools that serve different use cases and purposes. So at the very left, you have our Python and MATLAB APIs. They are very flexible and allow you to do very advanced things, but they require you to write custom code. At the center, you have NeuroConf, which provides more automation. So it's a low-code automated conversion for common input data format. It significantly reduces the amount of code that you need to write, but it still allows you to, with great flexibility, interact with the data. And then on the very right, we have Guide, which is a no-code automated conversion solution. So it allows you to interact with your data through a graphical user interface without requiring any coding. And so depending on your particular use case, you will pick tools from this spectrum and you will learn about these tools throughout this workshop. Right? And so first up is Guide. Guide you can think of as a TurboTax-like interface for converting your data to NWB. So you have a convenient interface that guides you step by step through the conversion process through a question and answer type process. NWB uses NeuroConf at its backend, and so you can now support 40+ different data formats that you can convert to NWB with Guide. You can conveniently enter your metadata in the graphical interface, so a step-by-step process, and that data is immediately validated and documented for you. You can then visualize and inspect your data before you run a full conversion. Even Guide uses NeuroSift for visualization. And so before you run a conversion of your entire data set, it can run a sample conversion where it just converts a small part of your data so that it can validate your data before you do large batch conversion through the interface. And then finally, once you've converted all your data, you can also upload your NWB files with Guide to the Dandy Data Archive for sharing. The other tool that I mentioned is NeuroConf. It's an automated NWB conversion suite. So NeuroConf allows you to convert data with minimal time and effort to NWB. It supports 44+ different data format. It automatically extracts specific metadata from your raw data, and it can seamlessly integrate your data. So it can convert very large data at terabyte scales and optimizes the data for streaming access from Dandy. And so through NeuroConf, with very few lines of code, you're able to convert your data, such as in this particular case, we have a Spike GLX converter pipeline where you're just specifying where your Spike GLX data is being stored. You're specifying your basic metadata for the NWB file, and then you just in a single call can run your conversion. So with minimal code, you can do your conversion from standard data formats while still being able to then interact with the file to do more advanced things through the custom APIs. Finally, we have our APIs, PyNWB and MatNWB. They're useful for anytime you have more custom needs and you need to dive deeper into NWB. It supports all the features of NWB, including trial times and responses and properties of those. They support iterative data conversion, so you don't have to load all your data into memory to do conversions, but it will load the data in chunks and be able to convert data at very large scales in that way. Similarly, it also supports lazy data load, so even for very large data files, you will be able to open them and load data on demand. It also supports chunking and compression and so on. And then finally, NWB Inspector is a tool that allows you to inspect your data for compliance with best practices and to check your data for errors. It is integrated with the Dandy upload process as well as with NWB Guide. So you will be using the inspector through Guide and the Dandy tools, even if you're not directly aware of it. You can obviously also run the inspector directly yourself to get reporting on your data if it's compliant with best practices. And then finally, once you have converted your data, you can upload it to the Dandy Data Archive. Dandy is the brain archive for publishing and sharing neurophysiology data. It supports storing of hundreds of terabytes of data for you for free, and it can handle both raw and processed data. It supports upload and embargo mode as well as publishing with DOIs. Okay, so now you have a rough idea of the different tools that you can use to get your data into NWB, as well as a high-level idea of what the NWB data standard is. But really, NWB is more than just a data standard and a set of tools. But NWB technologies are really at the heart of the neurophysiology data lifecycle. Data standards, including NWB, are really critical conduits that facilitate the flow of data throughout the data lifecycle, from acquiring the data to processing it, analyzing it, publishing the data, and reusing it. And so that means NWB needs to meet the needs and support the challenges across the different technologies that we need in this data lifecycle. And so a core value of NWB is that we're seeking to work with, not to compete with, existing and emerging technologies. And so as part of our work, we're engaging with tool developers and the community more broadly to integrate other technologies with NWB. And then finally, NWB is a data standard for neurophysiology data, not a standard of neurophysiology experiments, meaning we are here to help you share the data that you're generating, but our goal is not to dictate the sort of experiments the community is trying to do, but really to help you engage with the community more broadly. And so over the last few years, we have been able to really grow together with the community a broad ecosystem of tools that exist around NWB and support its integration. That ranges from general tools for data exploration, such as NeuroSift. There's a broad range of HDF5 tools that support reading NWB files. There's NWB widgets, NWB Explorer. All of those tools allow you to inspect your data and see what is going on in an NWB file, which data is available. There's also a broad range of data management tools that support NWB. So DataJoin, for example, is a pipeline management system that's popular in neurophysiology, and they're supporting, for example, export to NWB through their DataJoin elements. Spyglass is another interesting suite of data management tools that builds on DataJoin and integrates tightly with NWB to manage large collections of NWB files. I already mentioned Data Archive, and then there's more broadly positioned data analysis tools such as Pineapple and Zebra. And then there's a large collection of different tools for the different data modalities, such as extracellular electrophysiology, intracellular electrophysiology. There's a broad range of tools for optical physiology, such as Cayman, Cicada, Opnist, and so on. And then finally, there's also a growing range of tools in the behavior space, such as deep lab cuts, sleep, and others for doing pose estimation, as well as tools for doing controls of behavioral experiments, such as air control, beetle, and Visio mode. And so these tools provide you with a broad range of assets, really, to enhance your data analysis, to make your life easier as you're processing your data once you have your data integrated with NWB. Now, in this last part, I want to talk a little bit about extending the NWB data standard, and this is really targeting to enable the community to contribute to NWB and adapt NWB to the changing needs of the community. And so for that, we have to dive a little bit under the hood of NWB, right? I talked so far about data APIs and tools and interfaces that you will be using to interact with NWB data. I also talked a little bit about the data standard itself as a fundamental schema that governs the data standard. But to make it extensible, we also need to talk a little bit about the actual specification language that's being used to define the data standard, as well as how different data storage modalities integrate with NWB as a data standard, right? And so first of all, when we talk NWB as a data standard, it is governed by a formal schema. And that schema is defined using the NWB specification language. So the NWB specification language defines how data objects are organized in the data standard. And so there's different types of objects you will encounter: groups, which you can think of as folders; data sets, which are n-dimensional arrays; links and attributes. And so the specification language allows us to define their core properties, such as their names, which data types arrays have, which dimensionality they have, and so on. And so we then use that language to define a vocabulary of neurodata types. Neurodata types, as I mentioned, are components in the data standard that are reusable and extensible. So we have, for example, time series as a core data type. And then we can inherit from that time series to define more specific recording types, such as electrical series or optical series, that all act like a time series, but define more specific properties related to extracellular recordings or optical recordings. And then these neurodata types can also be composed by including them in other types. And so using that specification language, we can define new components for a data standard. Importantly, we can use the specification language to define our own extensions to the NWB data standard. And so NWB provides a broad range of tools to help you with that extension process. So first of all, the PyNWB API provides an interface for defining new schema. So you can programmatically, which is what you see on the left, create a new schema by saying, I want to create a new group. That group should contain a particular data set with a certain number of dimensions and data types. And so you can programmatically build up your extension and then export it, which will give you a YAML file with the formal schema. Once you have the schema for your extension, you can then immediately use it with the NWB file. So you can load that extension in PyNWB or in MetaNWB. It will then automatically create you classes to represent the data in your new schema. You can add that to your NWB file and save it directly. Importantly, these extensions are not just immediately usable, but they also become immediately shared directly with your data file. So as you share your data, others in the community will be able to then access the data also through the standard APIs, because the extension is directly being shared with the data together. And then NWB also provides a broad range of tools and a formal process to facilitate the creation of these neurodata extensions. There is a formal template that you can use to create new extensions. It provides you a step-by-step process for setting up new GitHub repositories, for creating the extensions and editing them. We have our neurodata extensions catalog. That one you can use to register your extensions, to publish it to the community. So the catalog stores metadata about your extension so other people can find it and use your extension, as well as to then start collaborating on these different extensions. And then finally, we have developed with our technical advisory board a formal process for NWB enhancement proposals, so that the community can use extensions to propose new enhancement to the core NWB data standard. And so through that process, you're able to develop new parts of the NWB data standard, really thoroughly test them with the community, collect feedback, and then once they're ready, we can make a proposal to have them formally integrated with NWB. And then finally, there's also the component of data storage. Right? The specification language and the schema define how data should be organized. And then the data storage component defines how we take those organizations and translate them to disk. As its primary data storage backend, NWB uses HDF5. HDF5 has the advantage that it allows us to collect all those different types of data in a single file that we can then share and publish. It supports large-scale data storage. It's optimized for performance and parallel I/O on large compute system. It is supported across all major platforms and languages, from MATLAB, Python, C++, Fortran, R, and others. And it targets long-term support, which is important for NWB. And it also supports advanced I/O optimizations, such as chunking, lazy data load, compression, and so on. And so as part of our efforts in NWB, we're also working with the community to explore integration of other storage technologies, such as about a year ago, we released HDMFZAR, which provides a ZAR-based storage backend for NWB. We're also developing HERD as a new tool to enable you to annotate NWB files with ontologies, as well as working with DataJoin on efforts such as DataJoin elements to allow you to integrate NWB with the DataJoin ecosystem. To learn more about NWB, you will find a broad range of online resources. NWB.org will tell you information about the broader organization of the project and our policies. If you're starting to dive into NWB to adopt it with your data, I highly recommend that you visit the NWB Overview website, which will provide you a guide to finding the different resources that you need. And then there's also a broad range of documentation associated with the individual tools. If you need to get help, reach out to us here while we're in the Gather space with you, but also you can reach out to us on Slack, as well as our GitHub help desk, and then for specific issues also through pull requests and GitHub. And with that, I am concluding my talk.